Welcome to clinical parasitology. The question is really, who cares about parasites? And most people have really never heard much about parasitology before. And it is incredible how it is so prevalent all over the world. Some of you may have watched the movies Monsters Inside Me, which is really pretty cool. And oftentimes it's a little bit scary. There was also a movie called Parasite, which was about human parasites, but in this class we obviously talk about all the different parasites that affects human, animals, and the environment. In the middle picture you can see the head or skull legs of a tapeworm, and that's a real image of how the parasite attaches to the intestinal walls. The cat on the right depicts the interaction of toxoplasma and the cat being the definite host of it, and we'll talk about that a little bit more. So again, most of you may not have a really big vision of what parasitology means, but it comes in many different forms and shapes and sizes. All right, so parasite, what does it mean? It's an organism that lives in or on another organism, the host, and benefits by deriving nutrients at the host's expense. We often make jokes as well that humans can be parasites as well, but that's not what the course is about. Parasitology has really played an enormous role in the evolution and changes of how humans have existed. Parasitology is important in religion. It's important in how cultures have evolved, and there's several really good books about this topic, and read on if you want to know more about it. Again, this course is more about the technical aspects and the diagnostics of parasitology. Alright, so symbiosis. Symbiosis means literally sym, together, bios is living. So organisms living together, and it comes in many different forms. Many organisms live together with mutual benefits. Commensalism, where one has benefit and the other one doesn't have any effect. And in parasites, where one exploits the other one and the host is usually harmed. Very common parasites are ticks. They transmit Lyme disease, other things. And on the top right, you can see mistletoe, which is a plant that lives on another tree. The bottom left is a fungus that exploits insects, that changes their behavior. And then you just have other parasites that feed organisms that feed on other organisms. There are really some great resources, videos, documentaries, TED Talks about parasites. So if you're interested, go into it. Again, this course is more about the technicalities in clinical microbiology. The human parasites are usually classified in 3 different groups. The protozoans, and again, we'll have chapters on each one of those. The helminths, the worms, and then ectoparasites. And they are the parasites either themselves, and oftentimes these parasites also transmit diseases. So, very important group. The protozoans, again, we'll have chapters on the different parasites. They're classified as amoebas, flagellates, sporozoans, and ciliates. Again, we'll get into much more detail on all of these. The helminths, the worms, are classified into flatworms, platyhelminths, roundworms, nematodes, and thorny-headed worms, acanthocephali. They're not very common. The last group is not very common, but I actually did find one when I was collecting fecal material from our pig facility at UF. I did find one in the pig facility. It was pretty cool to actually see them. It was my first time. Right, ectoparasites, again, lots of different insects mostly. And then you've got the leeches as well. And we'll talk about them in a lot more detail. One big topic about parasitology is the tropical neglected diseases, or TNDs, and there's a lot of information on at the World Health Organization. A lot of these diseases are affecting people in underprivileged countries, but there has been, over the last few years, they have made a lot of progress where some of these pharmaceutical companies have donated medication, and some of these diseases are actually now considered reduced to eradicated. So a lot of research is being done in this field, and lots of progress have been made as well, but still a lot more progress needs to be made in the future. Neglected tropical diseases can be controlled, and what I said, a lot of effort has been made. This is a picture of elephantitis, where the lymph channels are blocked, and that's why you had a lot of accumulation of fluids in different areas of the body. Again, it is quite common still, and it's a fairly easy one to be treated and eradicated, but a lot of people are still suffering, and you can obviously see that it affects people's lives from these diseases. Animal reservoirs are important, especially as we encroach on different ecosystems. We build houses, we build roads into the forests, and so there's a lot more contact between animals and humans, and that's where transmission often occurs. Most of our wild animals in Florida and beyond are riddled and are full with parasites, so getting close to them and interacting with their environment often has the consequences of transmitting diseases to humans. And yeah, so again, reservoirs, parasites exist everywhere, and lots of animals in our environment have parasites. So again, interacting with them risks the transmission of humans to parasites. Vectors. So vectors is an animal that transmits a disease, and we are obviously very familiar with insects that transmit diseases like malaria or sleeping sickness. The ticks, they transmit many diseases, including Lyme disease. Fleas, they transmit the plague, the bacterial disease, and Glossina that transmits sleeping sickness as well. So again, lots of different vectors that transmit diseases. They just, bite for blood meal, for survival, for their food, but at the same time they transmit other diseases. Again, lots of different vectors that can carry diseases and transmitted parasites and other diseases like bacterial diseases. The life cycle of parasites is obviously very, very complicated, and the co-evolution of humans and parasites has existed for millions and millions of years. This one is the transmission of sleeping sickness by the tsetse fly, and this was actually my PhD work. So I worked on sleeping sickness and some of the genetics underlying the resistance of humans to some of these parasites. One of the parasitic characteristics for some of the parasites is, and that's one of the reasons why there's very few vaccines against parasites, is they can change their surface. So antigenic variation is a genetic trait of parasites where they can change the surface so they look different in different generations. And the humans, obviously, they build antibodies to these parasites, and if they change the surface, the antibody can't recognize it. So this is an old paper by Ronald Ross more than 100 years ago, where he recognized that trypanosomes have a certain antigen at the beginning when they infect the humans, once the human mounts antibodies, the first generation of these parasites gets eliminated, and then the parasite changes, and humans produce other antibodies against the second type of parasite, and so it continues. But what happens is that the human antibodies always catch up, and the parasites wins because it can change the surface so many times. So that's where you get the cyclic nature of the amount of parasites, but it also correlates with fevers and disease symptoms as well. So scientifically very interesting phenomenon of antigenic variation, but in reality it's really difficult to overcome that and produce vaccines and immune systems to these parasites that can avoid the immune system so readily. Combating parasites is really difficult because you have the biology of the organism, you have the ecology, the environment, and especially now with changes in the environment, you get increased dispersion of vectors. And you also have the epidemiology, the host reservoir, and human behavior. So it's very complicated to eliminate parasites because there's so many factors involved, and most people don't really understand the interaction of the different variables. There's lots of different example parasites, and we will go into all of these different groups of parasites and explain them in these next chapters. So the introductory chapter is very, very dense. There's a lot of information on there. So read through the chapters and, get the highlights. We, in this class in clinical parasitology, the key focus is obviously to pass the ACP exam, and it focuses a lot on identification, on diagnostics, on image analysis, on fixation and staining. So we will go over all these methods in due time. And this was just a really quick introduction of parasitology